Hello guys, welcome to this YouTube channel. A good day to everybody watching this video. In this video, I'm going to discuss the answer key for the biochemistry section of the GATE Excel 2016 paper. So let's get started. The first question, the heterologous expression of green fluorescent protein, that is GFP, is possible because the genetic code is, the correct answer here is universal. Because the genetic code is universal, even in different organisms, the same genetic code can be used to express a given protein, say like GFP, which can be used as a marker protein or so on. Even though there are few exceptions, but mostly the genetic code is universal and is the same for all the organisms throughout the kingdom. Moving on to the next question. Phosphoglucose isomerase was incubated with 0.2 molar of glucose 6-phosphate. On reaching equilibrium, 55% of glucose 6-phosphate was converted to fructose 6-phosphate. The equilibrium constant for this reaction is, so first let's write the reaction. So here we have glucose 6-phosphate, let me write it as G6P, is getting converted to fructose 6-phosphate. Let me write it as F6P. And the enzyme that is doing this is phosphoglucose isomerase. Phosphoglucose isomerase. Okay. So here the reaction starts with point, let's say T equal to T0, which is the initial time. The reaction starts with 0.2 molar glucose 6-phosphate and there is no fructose 6-phosphate. And upon reaching equilibrium, let's call it TEQ, that is at equilibrium time, we see that 55% of this glucose 6-phosphate is converted to fructose 6-phosphate, which means this is 55% of 0.2 molar. And since this is 55%, the remaining 100 minus 55% will be the glucose 6-phosphate, which is 45%. So it's 100 minus 55. 45% of 0.2 molar. That will be the concentration of glucose 6-phosphate at equilibrium. Now we need to find the equilibrium constant for this reaction. So equilibrium constant is equal to K equilibrium is equal to product concentration at equilibrium divided by reactant concentration at equilibrium, right? This is basic chemistry that we know, inorganic chemistry. So that will be concentration of fructose 6-phosphate at equilibrium divided by concentration of glucose 6-phosphate at equilibrium, which is equal to 55%, which is 0.55 of 0.2 molar divided by 0.45 of 0.2 molar. So that will be equal to 1.22 approximately. It's 1.22, it's recurring, so it's 1.22. So the correct answer here is 1.2 as per the answer key. I hope this is a very simple question if you know basic inorganic chemistry. Moving on to the next question. Hydrolysis of a peptide involves cleavage of the bond between the atoms. What? So basically you should know what the peptide bond is. So it is CONH and then here you have. So here you have the C alpha and here you have the C prime alpha for the next amino acid. So here is the other amino acid. So here the hydrolysis occurs here. So it is the cleavage occurs between the N and C. So the correct answer here is option D, which is N and C. This is a pretty simple question. Moving on to the next question. Interconversion of UDP glucose and UDP galactose is catalyzed by. So first of all, here you don't have to worry about what UDP is because that group is unchanged between the product and the reactant. However, you need to know the relationship between glucose and galactose. So glucose and galactose are epimers. It is very important that you need to know what type of epimers they are, whether they are C2 or C4 and so on. So it is important that you know the examples of C2 and C4 epimers with respect to glucose. Anyways, glucose and galactose are epimers. So hence this interconversion is catalyzed by an epimerase. So the correct answer here is option C. Moving on to the next question. So here, the gel filtration profile and the corresponding activity, let me just zoom in here. The gel filtration profile and the corresponding activity data for a pure enzyme are shown in the figure below. The same enzyme sample on SDS page runs as a 30 kilodalton polypeptide. So now they have given this figure here, and here the first purification is done, and then in SDS page it runs as a 30 kilodalton polypeptide. Okay, so which one of the following is the correct interpretation of the data? So first of all, you look at the absorbance, that it is higher at 60 kilodalton, and also the absorbance is high at 30 kilodalton, which means the enzyme is basically getting cleaved here from 60 kilodalton to 30 kilodalton. Probably it's a dimer. And however, you also observe that the enzyme activity is really low at 30 kilodalton. It's almost zero. However, it increases when it is at 30 kilodalton. So here the correct answer here is option B, that is enzyme is active only as a monomer. So it's a dimer when it is at a 60 kilodalton. However, when it is a monomer, that is in terms of 30 kilodalton, it is the enzyme activity is really high, as seen in the total enzyme activity unit here. So the correct answer here is option B, that is enzyme is active only as a monomer. So this is a very simple interpretation that you can do from the given graph. Moving on to the next question. Amino acid residues predominantly involved in protein-DNA interactions are. So basically DNA is negatively charged. Why? Because of the phosphate backbone, sugar phosphate backbone, and definitely the proteins that are involved in protein-DNA interactions are positively charged. So the correct answer here is positively charged. Okay. Moving on to the next question. So cellulose serves as a structural polymer, whereas starch does not. This is because cellulose contains. So basically cellulose has beta-1,4 linked glucose monomers, which is like the branching that you can see, and there is also interchain hydrogen bonds, not intra. So interchain is between two chains, whereas intra is within the same chain. So since cellulose serves as a structural polymer, it has the branching due to the beta-1,4 linkages between the glucose monomers, and it also has these interchain hydrogen bonding, and hence it is a structural polymer. This is a simple question. Moving on to the next question. So here the molar absorption spectra labeled 1, 2 and 3 for 3 different amino acids are given below. So clearly this is the molar absorption spectra for 260, 280. If you can see, it's between the aromatic amino acids, so we know that, which is also used for determining the concentration of proteins based on the Beer-Lambert's law. And which one of the following is the correct combination of the spectral assignment? So here 3 is definitely phenylalanine because it doesn't show an absorption maximum at 280. So 3 is phenylalanine, so it should be either option A or option D. And the main absorption mainly comes from the tryptophan, that is 1. So you can see that the molar absorbance is really high for 1. So 1 corresponds to tryptophan, 2 corresponds to tyrosine, and 3 corresponds to phenylalanine. And hence the correct answer here is option A. So this is a pretty simple question, basic biochemistry question that everybody is supposed to be knowing. Moving on to the next question. The fluidity of a phospholipid membrane increases when? So here the fluidity means that the chain length decreases and the degree of unsaturation increases. So when the chain length decreases, the hydrophobicity is also low, and when the degree of unsaturation increases, the fluidity also increases, which means it's pretty easy to move. So that's how the fluidity of the phospholipid membrane increases. And I would also request everybody to know under what temperature conditions and the degree of unsaturation, chain length, there are different parameters that actually determine the fluidity of a phospholipid membrane. And I would suggest everybody preparing for GATE Excel biochemistry or any biochemistry related competitive exams to know how different parameters affect the fluidity of a phospholipid membrane. Moving on to the next question. So here the polypeptides are biosynthesized on ribosomes inside the cell. Chemical synthesis of polypeptides is also possible through Merrifield solid phase peptide synthesis. In both the cases, the polypeptide chain is extended 1 amino acid at a time. Yes, the direction of polypeptide synthesis is. So here, the correct option here is option D. That is, it is from N-terminal to C-terminal on the ribosomes. That's like the normal way, even the conventional way we read the peptide, the amino acids in a peptide chain. However, it is the opposite in case of the solid phase peptide synthesis, that it is from C-terminal to N-terminal. If you remember from my previous videos, this question was also repeated in one of the recent GATE Excel examination. So it is really wise that if you could go through all the previous year's question, it is also possible that in the upcoming exams, any question might even get repeated. So it's always a good idea to practice previous year's questions. And this particular question was repeated in one of the recent Excel papers, and that could also be seen in my previous videos. The correct answer here is option D. Moving on to the next question. So here the 4 groups of metabolites are given below. So each group has 3 metabolites. Okay, choose the group in which all the compounds contain at least one bond whose delta G naught, that is standard free energy of hydrolysis, is less than minus 7 kcal per mole. So basically they are high energy bonds. So here the correct option here is answer B. So here if you look at the options, the metabolites here, that is creatine phosphate, acetyl phosphate and succinyl CoA, all of them have at least one bond whose delta G naught is less than minus 7 kcal. However, if you look at the other groups, there is there's always one which doesn't have this, which doesn't have a bond that is less than minus 7 kcal. They are pretty stable compared to the other metabolites. So in this particular group, all 3 metabolites have at least one bond that has a delta G naught value less than minus 7 kcal per mole. So the correct option here is answer B. Moving on to the next question. The delta G naught for the malate dehydrogenate catalyzed in the catalyzed step of Krebs cycle is plus 7 kcal per mole. Nevertheless, the conversion of malate to oxaloacetate in vivo proceeds spontaneously because the subsequent reaction that consumes oxaloacetate has a delta G naught of. So basically here it's going to be a coupled reaction. So if the coupled reaction is the one that is resulting in the final reaction, then the second reaction needs to have a delta G naught that is more negative compared to the given one. So in this group of option, the correct answer here would be option C, that is minus 7.7, because minus 7.7 is more negative than plus 7.1. That's obvious. As a result, if you combine the 2 delta G naught together, you will get minus 0.7, and that is making this reaction spontaneous in vivo without the need for any further steps. So that is the reason why the correct answer here is option C, because here the conversion of malate to oxaloacetate in vivo is spontaneous. However, even though it is plus 7.1, the coupled reaction results in an overall negative delta G naught. And that is possible if the second half of the reaction is more negative than plus 7.1, and the only option that matches this is minus 7.7. That's a pretty logical question. Moving on to the next question. When freshly isolated intact mitochondria were incubated with ADP and inorganic phosphate, neither the oxygen consumption nor the ATP synthesis could be detected. Addition of succinate resulted in increased oxygen consumption as well as ATP synthesis with time. So subsequent addition of cyanide to the system will result in which one of the following? The correct answer here is option A. When you add cyanide, both the oxygen consumption and ATP synthesis are inhibited, so which completely blocks mitochondrial respiration and even ATP synthesis. So the correct answer here is option A. Moving on to the next question. So here, 3 micrograms of a circular plasmid of 4200 base pair was digested with a restriction enzyme and subjected to agarose gel electrophoresis. 5 DNA fragments of different sizes were observed and their sizes summed up to 4200 base pairs. Yes. So the number of picomoles of DNA ends, be very careful with the words here, they have asked DNA ends, generated after complete digestion with the enzyme is what? And they have also given another information, that is the average molecular weight of each base pair is 660 Dalton. Okay, so first of all, let's draw a plasmid to make it easy. So in order to generate 5 DNA fragments, we need to have 5 restriction sites, right? So this gives you 1, 2, 3, 4, 5. So when there is going to be 5 restriction sites, there's going to be 2 DNA ends that is going to be generated here. So there's 1 DNA end here, 1 here, then there is 1 more here, and then there is 1 more here, 5, 6, 7, 8, 9, and 10. So there are going to be 10 DNA ends that is going to be generated per plasmid upon restriction enzyme digestion. So first of all, we should calculate how many plasmids are there. So to do that, we need to calculate from the given data. So the number of plasmids would be equal to, number of plasmids is equal to 3 micrograms, which is 3 into 10 power minus 6 gram, divided by 4200, which is 4200 base pairs. Each of them is 4200 base pairs into 660 Dalton. So each plasmid weighs about this much. So 660 Dalton. So what is Dalton? Dalton is gram per mole. Okay, so let's keep it as per mole because they have asked the answer in picomoles. So 660 grams per mole. So this is 3 to 10 power minus 6 grams divided by 4200. That's the total number of base pairs in the plasmid into 660 gram per mole. So this gives you the number of plasmids in terms of the number of moles. So when you calculate this, you get the answer to be approximately 3 by 2.77 picomoles, because I just converted this to picomoles because the final answer was asked in picomoles. However, this is for the number of plasmid, and each plasmid has 10 DNA ends. So now the total number of DNA ends would be equal to total number of DNA ends will be equal to 3 by 2.77 into 10 picomoles. So that will be 30 by 2.77, which is approximately 10.8. So the correct answer here is 10.8 picomoles. So that's the number of DNA ends that are generated after complete digestion with the enzyme, restriction enzyme. So this calculation I didn't show in detail because this can be done using a calculator, and I've just kept this as picomoles, and I've kept this as a fraction just to simplify my calculation, so that if I multiply by 10 and then finally divide it, it's going to save the number of steps. Moving on to the next question. So here, an enzyme was purified using ion exchange chromatography, and the results are shown in the table below. So here is the table, and we need to figure out the purification and yield so that the correct answer can be matched. So purification, so before we find purification, we need to also find specific activity. So specific activity is equal to total activity divided by total protein. So that gives you specific activity of the enzyme. So here, these are like different steps of the purification. This is the first step, this is the second step. So here, the specific activity is 800 divided by 400, which is 2. And here, the specific activity is 200 divided by 2, which is 100. So here you divide these two. So specific activity is total activity divided by the total protein. And now, we need to calculate purification. So how will you calculate purification? Purification is equal to final specific activity, or the specific activity in the final step, final specific activity divided by initial specific activity, right? Final specific activity divided by initial specific activity. So what is the final specific activity here? That is 100. So 100 divided by 2. So it is 50-fold purification. Okay, so that is found. Now we need to find the yield. So yield is equal to final total activity, or the total activity in the final purification step, final total activity divided by initial total activity, into 100 if you have to express it in percentage. So that will be equal to 200, which is the final step in the purification here as shown in this table. 200 divided by 800 into 100. So which is 200 divided by 800 into 100, that is 1 by 4 into 100, which is 25%. And hence the correct answer here is option A, which is 50-fold purification was achieved with 25% yield of the enzyme. So again, it's not a very great purification, but still this is how you calculate. Moving on to the next question. So here, the aspartate residues are found in the active sites of many enzymes. The pKa for the beta carboxylate of aspartate is 3.86. At physiological pH, this group can function as. So first of all, you should know what the physiological pH is, which is 7.4, right? So this 7.4 is greater than 3.86. If you look at the Henderson-Hasselbalch equation, which says that pH is equal to pKa plus log of salt by acid concentration, here you find that this pH is greater than pKa, because pH is the physiological pH and pKa is 3.86. So which means that the salt concentration is greater than acid concentration, right? Because this term is positive, which means that salt is greater than acid. So if this is going to be the case, then this particular beta carboxylate group is furnishing out its H plus ions, which means that it is existing as COO minus. So when it existing as a COO minus, it will function as a nucleophile because it is rich in negative charge and as a conjugate base. So when it furnishes H plus ions, it gets converted into a conjugate base. So the correct answer here is option C, which is a nucleophile and a conjugate base. This is basic thing. Moving on to the next question. So here, the kinetic parameters for the enzyme Fumarase with 3 different substrates are given below. So you have 3 substrates, that is fluorofumarate, fumarate and chlorofumarate. So the specificity of Fumarase, which is the enzyme, for the substrates decreases in the order. So the specificity is determined by Kcat by Km. So here, if you look at this, the higher the Kcat by Km, the higher the specificity for an enzyme, for a given substrate. And if you look at this, we can clearly say that 800 divided by 5 is greater than 2700 divided by 27, which is greater than 20 divided by 111. So 800 divided by 5 is for fumarate, right? Yeah, fumarate, which is greater than for 2007, which is greater than fluorofumarate, and this is greater than chlorofumarate. So the correct answer here is option C. That is fumarate greater than fluorofumarate, which is greater than chlorofumarate. So fumarase has very high specificity for fumarate compared to fluorofumarate, which in turn is compared to chlorofumarate. So that's a simple question. Moving on to the next question. So here, a polypeptide with amino acid sequence, this is a sequence, it's a small peptide, was dissolved in a buffer of pH 1.8. The predominant form of the polypeptide will have a net charge of. So let's first write this polypeptide. So AGKPDHEKAHL. Always write this is N-terminal. When it is not given, it is safe to assume that this is the N-terminal and this is the C-terminal. Okay. So here, first of all, we need to mark the amino acids which are positively charged, so that they also contribute to the positive charge, because it's a very low pH, which is 1.8. So lysine is 1, so you have one positive charge here. And another one is histidine, you have another positive charge here. You have another lysine here, which is another positive charge here. And you have another histidine, you have another positive charge here. And alanine, being at the N-terminal end, will also be positively charged because it's a very low pH, and the NH2 group will be protonated by another hydrogen ion. So here this will also be positively charged. And here leucine, which is at the C-terminal, will remain as COOH instead of COO minus. So that will be neutral. There won't be any charge here. So when you add these, you get plus 5. So if you know the positively charged amino acid residues along with including another extra charge for the N-terminal end, because it's going to be NH3 plus, then you will be able to solve this question. So the correct answer here is option B. Moving on to the next question. So here, an N-terminal his-tagged protein of molecular weight 40 kilodalton was purified using NINTA column. This protein sample was subjected to SDS page. A Western blot of the same using antihistidine antibodies is shown below. So which one of the following interpretations is correct? So here it was initially a 40 kilodalton protein which was his-tagged, and during Western blotting you use these antihistidine antibodies, and when upon running these SDS page we also find a 20 kilodalton which was found. So this means that the protein has a stable N-terminal 20 kilodalton domain, because here it says that the his tag was N-terminal tagged. So since you also see a 20 kilodalton fragment, it means that this protein has a stable N-terminal 20 kilodalton domain, and since you get this blot only upon these antihistidine antibodies binding to this protein, it implies that this protein has a stable N-terminal 20 kilodalton domain. So the correct answer here is option D. Moving on to the final question. So here, the sequence of a polypeptide that forms a transmembrane helix is shown below. So it's a big polypeptide. Which one of the following segments of the peptide is most likely to span the membrane? So here, there are many methods by which you can solve this question. However, I prefer, going with the deduction method because sometimes it can be really confusing, especially when you have these sliding window theorems and stuff like that. So the way in which I would go is look at the options and figure out which of these groups have the maximum number of hydrophobic amino acids or neutral amino acids. So because this is going to span the membrane, this has to be hydrophobic or neutral in charge, or these amino acids should have a neutral or hydrophobic side chain. So if you go by that approach, the correct answer here would be option C, because here it is from E15 to A34. That is from here to A34. That is this. If you look at the amino acids here, that is isoleucine or threonine or leucine and isoleucine, phenylalanine, these are all highly hydrophobic and they span the membrane and they can easily span the membrane. However, if you look at the other options, like for example E3 to G22, that is this, you have a lot of these positively charged and negatively charged, like Q, which is asparagine or glutamine, and you have histidines here, and then you have that is aspartate here, and then these are all like positively and negative. These are all charged amino acids, and likely they cannot span the membrane. Similarly, if you look for V5 to V25, also you will encounter these, and similarly for F21 to R40 also you will encounter these. So if you go by deduction method, it is easy to do. However, there are also many theorems that can actually help you to solve this, but I prefer doing through this particular way because it's pretty easy to avoid mistakes. So the correct answer here is option C. So with this, I'm completing the discussion for the answer key for the GATE Excel Biochemistry section of the 2016 paper. I hope you found this video interesting, and I also hope you found this video useful, and I wish everybody a good preparation time, and also I hope that all of you ace the upcoming competitive exams in biology. So have a nice day. Thank you. Goodbye.